So let's see if I can give you some more perspective on how to use that NCBI genome viewer. In here. So in this case, we searched some gene. The gene was called the DRD5, and then we clicked on the result and end up in this page. So on the top, it's giving you an overview of what is encoded in this genomic DNA sequence. So these are the numbers of, let me get it bigger. This is the number of the nucleotides in this position, and then as you see, it goes up. In this view, this is always going to go up, and then down here, these numbers are going to go down depending which gene you're looking at. So since you're looking at this gene that is also going from left to right, then this viewer doesn't have to flip your view. So we are focusing on this gene. So these numbers are going to go from lower to higher. So those are the nucleotide numbers for that position. If you think about these are as one street and then these are house numbers, then you are going, maybe this is a boulevard, okay? So you are looking, you can either drive in one direction, the boulevard, or on the other direction. So we are driving on the direction of the boulevard that the numbers are going up, okay? So when we want to find out more information about this one gene, then we're going to look at the green track. The green track is showing you the DNA sequence. If I expand the green track, then I will see the mRNA sequence and the protein sequence. And when you expand that to see the mRNA and the protein sequence, then it becomes clearer what the structures, the parts of the gene are. So for example, right now we see light green, dark green, and you don't have to memorize what that is. I mean, it's easier if you memorize that dark green is coding and light green is the UTRs. But if you don't memorize that, but if you remember what UTR is, you could figure out by clicking on it. So we could look at the gene either on this one green track or this other green track. And so like this one or this one. And you might wonder what is the difference between them. And maybe you're like, oh, they are the alternative splicing. Not in this case. There was a case we looked at when it was alternative splicing. How do I know it's not? So this first one, this first green track, is the annotated from this project here, the main project. This other green track here is the annotation from the NCBI annotation release. So what this is saying is that when two different projects are annotating the genome, they agreed on the position of that gene. Just to keep things consistent, and because the NCBI annotation is the one that you most often find, then the questions I ask are going to come from the NCBI annotation. So you could keep this here or you could exclude it. I'm going to exclude now just so that it's a little easier for you to see things. So let's see. Oh, actually, there's an easier way to exclude it. I just want to not have that main project. Okay, so now I have the NCBI annotation. And they're like, okay, this is the gene I'm interested in because that's the one in the question. And then the arrows pointing left are confusing because they're like, isn't this gene encoded in that Watson strand? As in, we saw that it's pointing that way. That means that it's encoded on this strand. That means that the numbering should go up as it does here. If I was looking at this other gene, then these numbers would be flipped. They would be going down because this program flips it for you so that it's easier for you to see So it's like you're going on the other side of the street, where if you're going up the street, the numbers are going down. So the arrow pointing right means that you're on the side of the street, or you're on the strand where the count of nucleotides is going up. The arrows pointing left, you're on the other side of the street, you're on the other DNA strand, the numbers are going down. So normally this doesn't happen, that you have a DNA, some gene, and then you have some other gene at the same genomic location. I picked this for homework because it was really weird, and I wouldn't pick this for our exam because I don't want you to panic, especially if it would be your first time looking at it. Then you're like, oh my gosh, I've never seen this. What the heck? What is this craziness? But for homework, it's okay. So let's see. Yeah, so this is the gene that we're studying. This other one is a different gene. And if you're like, what is this? You could just hover over, and then you tell you the name of the gene. It's a different one. It's a solute carrier family 2, whatever. So it's something else. So that sequence does something else because it's reading the other DNA strand. And they're like, okay, I don't care about this. Let me focus on this gene here. And this is another annotation. That's the European one. That ensemble is the European one. And I'm going to close it, not because it's unimportant, but just because I want to focus on the NCBI one, because that's the acronym I want you to remember. That's NCBI. So let's look at this gene. If I click on it, then the tracks expand. So it always will show you the purple, which is the mRNA. And if you don't remember, then you just look here. It will tell you that's the mRNA. The red, you don't have to remember. You hover over it, and then it says CDS. Your job is to remember CDS stands for in genomics. That's coding DNA sequence. And then your job is also to know what does coding DNA sequence mean. So that's the DNA sequence that after it's transcribed, that transcript will be translated. So if there were introns here, or if there were introns here on the DNA sequence, then the introns in the red bar would be represented by skinny lines. This gene specifically does not have introns. This other one does. That's the skinny line for the other gene. So if I click on it, oh, it won't let me click. Okay. I wanted to show you that this would be represented by a red line, like a skinny line, because that would be an intron, so it's not part of the coding DNA sequence. Okay, so that's what's going to end up in the protein. This part here is the part that is the mRNA that is produced. So remember that the gene is the DNA sequence that is transcribed. So that green part, that green sequence gets transcribed, makes this molecule of mRNA, and out of this whole mRNA, only this chunk is going to be translated. So remember, the mRNA is not all coding. The parts of the mRNA that are non-coding are the introns, the 5' UTR, and the 3' UTR. So here's the 5' UTR of the mRNA, and here's the 3' UTR. And then let's see if you click on it. Now it's just going to collapse. So to get information, just hover over, and then this screen shows up. And let's see what else. So I want you to use this to learn the structure of the gene, and also to see, okay, what are the variations that we know of? And the variations we are looking at are the SNPs. So there's a database of SNPs, and it's saying, oh, we've sequenced this DNA, and we have reported that this position specifically can be a C or A or a T. And then this position specifically, this one position of base pair, can be any of these four. So that's what these guys mean. So the RS is the reference SNP. and then every SNP receives a number. And if you wanted to find out more about that SNP, you could hover over and it will tell you, and sometimes you can click on it. It tells you exactly the position on the genome. But the ones that I find interesting are the SNPs that show up as like little boxes like this, because then we know that more information about it. And sometimes they can be the pathological ones. So they have some clinical importance to them. So here these are clinical also, but this view doesn't show you which ones are pathological or not. And that's why I like adding this track that I told you. I'll tell you the name so that you can find it, because then it shows you the SNPs in little squares, and then you can see what's the clinical variation of it. So if I hover over this, it tells me that's that SNP. This SNP will also be on this. So here it is in here, right? Is it the same? It ends with 91. Yeah, so this is the same as this. But in this view here, it doesn't look special, right? And then in this view here, you'll be able to click on it and find more information. You could click on it here too, but it doesn't take you to the next screen. Or you can click on this, and that's going to end up in the same spot as if you clicked here. So there are different ways of going about it. The reason I want you to add this track is because it's easier to spot these tips that I want you to look at. Imagine that I gave you this view here, and then you didn't have this either. And I ask you, which one of these has clinical relevance? Then you have to click on each one of them. And I wouldn't do this to you. That's like way too much work. You'd have to like click on each one of them and see what happens and see if it's pathogenic or not. But that other view that I just closed shows you whether or not it's pathogenic.